Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic DNA replication. We'll see the different types of RNAs. So this is from the chapter Molecular Basis of Inheritance. So watch the entire video lecture to get basics out of it. So without any delay, let's start with the topic. So let's start. So let's see the different types of RNA. You can see on top the figure is given, that is mRNA. It's also called messenger RNA. Next you can see rRNA, that is ribosomal RNA, and next we can see you can see transfer RNA, that is tRNA. Okay, so RNA was the first genetic material. It acts as genetic material and catalyst. So essential life process. Okay, so it is essential for life processes like metabolism, translation, splicing, okay, etc. These are involved around the RNA. So DNA is evolved from RNA for stability. Okay, so DNA evolved from RNA for stability. So you can see the nitrogen bases. So three different types of RNA. One is mRNA, that is messenger RNA. Next is ribosomal RNA, and third is transfer RNA, that is tRNA. So we'll see about central dogma. What is a central dogma? You can see the DNA is converted to mRNA and mRNA is converted to protein. So whenever DNA is converted to mRNA, it's called transcription, and when mRNA is converted to protein, it's called translation. Okay, then DNA can do DNA replication. DNA gets converted to another DNA, that's DNA replication, and RNA get converted to another RNA, that is RNA replication. We can also know the reverse transcription, that is RNA to DNA, by the help of reverse transcriptive enzyme. So this entire process is called a central dogma. So you can see a central dogma of molecular biology. So it was proposed by Francis Crick. Okay, so central dogma was proposed by Francis Crick, and it states that the information flows from DNA to RNA to protein. Just now I've said, okay, you can see DNA gets converted to mRNA. Okay, RNA means the types of mRNA is mRNA specifically. So DNA get converted into mRNA, that is transcription, and RNA gets converted to protein, that is translation, and again DNA get converted to another DNA, that is replication. Okay, so this entire process is called central dogma. You can see the structure of DNA, the structure of mRNA and protein. Now, in some viruses, the flow of information is in reverse direction, that is in RNA to DNA. As I have said, in certain viruses, okay, there is a reverse transcription going on. That means we know the normal sequence is DNA to mRNA and mRNA to protein, but here you can see there is a reverse transcription. That means from RNA the DNA is produced. Okay, so you can see here DNA gets converted to another DNA, that is replication. You can see the reverse transcription is highlighted. You can see when RNA, when from RNA a DNA is produced. Okay, so that means DNA is formed from RNA, that is reverse transcription, and when RNA is produced from DNA, that is called transcription, and protein is formed from RNA, that is translation. So you can see DNA converted to RNA, that is transcription, and RNA gets converted to protein, that is translation. Here specifically RNA means mRNA. Okay, and the reverse process, you can see when from RNA DNA is produced, that is reverse transcription, and the replication is DNA is get converted to another DNA. So you can see HIV, it undergo reverse transcription. The HIV does reverse transcription, and this process is reverse transcription, and enzyme is reverse transcriptase. So let's start the topic DNA replication. Okay, so replication, what is replication? Replication is the copying of DNA from parental DNA. Okay, so replication is the copying of DNA from parental DNA. So Watson and Crick proposed, okay, semi-conservative model of replication. So inside our body we have semi-conservative mode of replication. It suggests that the parental DNA strands acts as a template for synthesis of new complementary strands. Okay, so after replication, each DNA molecule would have one parental and one new strands. So Messelson and Stahl in 1958 proved semi-conservative model. Okay, so you can see the two scientists. So Messelson Stahl's experiment, what they have said, they prepared two culture media of E. coli. One preparation contained NH4Cl, that is 15 nitrogen. Okay, so the NH4Cl salt, that is N15, that is heavy isotope of nitrogen. So N15 was incorporated in both the strands of bacterial DNA and the DNA became heavier. Okay, on the other hand, other preparation contain N14 salts. Okay, so N14 was incorporated in both strands of DNA and became lighter. So N15 was heavier and N14 was lighter. So these two types of DNA can be separated by centrifugation. Okay, in a CsCl, cesium chloride density gradient. So you can see the densities. One is N15, another is N14. N15 is heavier and N14 is lighter. Okay, so they took E. coli. What is E. coli? It's a bacteria, that's prokaryotes, Escherichia coli. So they took E. coli cells from N15 medium and transferred to N14 medium. Okay, N14 or 14N. So after one generation, that is after 20 minutes, they isolated and centrifuged the DNA. Its density was intermediate, that is hybrid between 15N DNA and 14N DNA. This shows that in newly formed DNA, one strand is old, that is 15N type, and another strand is new, that is 14N type. This confirms semi-conservative mode of replication. Okay, so one is N15 and this N14 in one particular strand of DNA. So in a DNA there is a double helix. So one strand is N15, another is N14. So that this type of replication is called semi-conservative mode of replication. So you can see after two generation, that is after 40 minutes, there was equal amount of hybrid DNA and light DNA. Okay, so you can see the pictures given. So whenever N15 DNA was taken, you can see after 20 minutes, you can see the semi-conservative mode of replication is seen, that one DNA helix, that is two strand having N15 and N14. But in the next generation, that is in after 40 minutes, you can see two parental strands DNA and two hybrid strands DNA. So in this hybrid strand, it contains one strand of N15 and another strand of N14. So Taylor and colleague in 1958 performed similar experiments on VCF-AVA, okay, using radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome. So it proved that DNA in chromosome also replicate semi-conservatively. So you can see this in VCF-AVA. So let's start with the replication process. Okay, DNA replication. So we'll see here the different enzymes that are involved and how the DNA replication takes place. So DNA replication starts at a point called ORI region. Okay, that is ORI means origin, and the unit of replication with one origin is called replicon. So what is replicon? A unit of replication with one origin. So during replication, the two strands unwinds and separate by breaking hydrogen bond in presence of an enzyme called helicase. Okay, so here helicase is an enzyme. So during the replication, the two strands unwinds and separates by breaking the hydrogen bond. Okay, so you can see the helicase enzyme is there. So unwind of the DNA molecule at a point forms a Y-shaped structure called replication fork. So whenever there is a Y-shaped structure formed, that is called replication fork. So you can see the origin of replication pictures is given, and the helicase enzyme picture is also given. You can see. Okay. So in this process of replication, there are three processes. One is initiation, elongation, and termination. Okay. So the separated strands acts as template for synthesis of new strands. So DNA replicates in which direction? It always replicates from 5' to 3' direction, because we know that in a double helix structure of DNA there are two strands. One is 5' to 3' and another is 3' to 5', that is antiparallelly situated. So deoxyribonucleotide nucleoside triphosphate. Okay, so deoxyribonucleoside triphosphate, that is DATP, DGTP, DCTP, and DTTP. That means deoxyadenine triphosphate, guanosine triphosphate, cytosine triphosphate, and thymidine triphosphate. So these acts as a substrate and provide energy for polymerization. Okay, so you can see the replication fork is growing. Okay, so you can see here the structure in the right hand side. You can see the chromosome. Inside chromosome there are DNA. Inside DNA there are the nucleotides, you can see. Okay, so firstly, a small primer is synthesized in the presence of an enzyme primase. So there is an enzyme called primase. It synthesizes RNA primer. So in presence of an enzyme, DNA dependent DNA polymerase, many nucleotides join with one another to prime a strand and form a polynucleotide chain, that is a new strand. Okay, so you can see the pictures given. So during replication, one strand is formed as a continuous stress, that is 5' to 3' direction, that is a continuous synthesis. This strand is called leading strand, and the other strand is formed in small stress, that is Okazaki fragment in 5' to 3' direction, that is called discontinuous synthesis. Okay, so here RNA polymerase is also there. So you can see the lagging strand and leading strand. The pictures is given. So the Okazaki fragments are then joined together to form a new strand by an enzyme called DNA ligase. Okay, DNA ligase joins, and the new strand is called lagging strand. Okay, so if the wrong basis is introduced in the new strand, that is a DNA polymerase can do proofreading. Okay, so E. coli completes replication within 18 minutes. So E. coli completes its replication within 18 minutes, that is 2000 base pair per seconds. So in eukaryotes, the replication of DNA takes place at S phase of cell cycle. Okay, so again I repeat, in eukaryotes the replication of DNA takes place at a S phase of cell cycle. Okay, so in a cell cycle there is a interphase and mitotic phase. Interphase includes G1 phase, S phase, and G2 phase. Okay, so in the S phase this DNA replication takes place, and failure in cell division after DNA replication results in polyploidy. So when there is a failure of DNA replication, it leads to polyploidy. So you can see in replication in bacteria, you can see the diagram, origin of replication and the replication process in both the direction. So you can see the old DNA and the new DNA formed. So replication is just a Xerox copy of the previous or the parental strands. Okay, so you can see the cell cycle, that is interphase and M phase. So interphase includes G1 phase, S phase, and G2 phase, and there is a M phase also, there is mitotic phase. So there is also a G0 phase where the cell division is stopped. So these are the machineries and enzymes. Okay, you can see here. Alright, so in a DNA replication, numerous enzymes are used. One is topoisomerase, helicase, primase, RNA polymerase, DNA ligase. Okay, so the following enzymes are used, and ultimately one DNA is converted to another DNA. So this was all about DNA replication. So thank you, student. This was all about the entire video lecture. We have discussed about the different types of RNA and DNA replication. So thank you, class.